Cell staining is a microscopy technique used to enhance the contrast of cells and their components, making them more visible and allowing for detailed visualization of structures like nuclei and organelles, aiding in fields like medical diagnostics and research. TCI offers a wide range of products suitable for cell biology studies. Cell fluorescent staining. Fluorescent staining techniques are essential tools for studying cellular structure and function for various cellular components. First, let's check out cell membrane staining. The cell membrane acts as a barrier between the cell and its environment, regulating material exchange and signal transmission. TCI offers lots of carbocyanine dyes, which are highly lipophilic fluorescent dyes. Carbocyanine dyes with long-chain hydrocarbons show little fluorescence in water, but they become fluorescent when incorporated into lipids such as cell membranes. Taking advantage of this property, carbocyanine dyes are widely used as membrane probes to study cell membranes. Here are maximum excitation wavelengths and maximum emission wavelengths. Examples of HeLa cells stained with each staining solution. PKH26 solution, Di-O solution, Di-R solution, Di-I solution, Di-D solution. Users can choose the appropriate dye according to the wavelength. Staining living and dead cells. Staining cells is an essential experiment for drug efficacy evaluation and toxicity testing. TCI offers dyes for staining live and dead cells. Staining in live cells. Calcium AM solution in DMSO is a reagent used in live cell staining. Staining in dead cells. Propydium iodide and PI solution are useful for identifying dead or dying cells in a population. In this sample, K562 cells were stained by calcium AM and PI. Green cells are living stained by calcium AM, and red cells are dead stained by PI. Lipid staining. Lipids play key roles in cell signaling, membrane structure, and energy storage. Let's see the in vivo imaging of lipid droplets with DBC30 or PC6S. In this example, you can see the images of the surface of the mouse liver. A. Healthy mouse. B. Fatty liver model mouse. DBC30 and PC6S stain lipid droplets in both cells and tissues clearly and distinctly due to the high fluorescence intensity. Additionally, TCI offers Nile red, Shoyaku green, pyromethane 546 for lipid droplet imaging. Let us take a closer look at the snippet of the product lineup of cell staining dyes from TCI. Methylene blue solution for cell staining. Oil red O for fat staining. Acridine orange solution for nucleic acid staining. TCI offers kits for mitochondrial isolation, mitochondrial membrane potential assay, nuclear or cytoplasmic fractionation. TCI also offers protein extraction buffers for E. coli or yeast cells, nervous tissues, and mammalian cells. Please contact TCI for more information.